Well, hello, everybody. Welcome back to another episode of Betterbee's Interviews with Experts series, where I sit down with people who know a heck of a lot more than I do about various topics, and we share some of that information with you. So today, I'm very pleased to be joined by Maggie Gill, who is joining us from all the way across the ocean to talk a little bit about Tropilaelaps mites. So hello, Maggie, welcome, and I'm looking forward to the chat.
Yeah, hi David. Yeah, thanks for inviting me along. It's exciting to kind of talk to you and talk about the research.
Yeah, excellent. So I thought we'd maybe just begin with your background. You know, what is your position? How is it that you come to be an expert on Tropilaelaps mites?
So I'm based in the UK. So I work for the UK government. I currently work for DEFRA, so Department for the Environment, Food and Rural Affairs. I'm a senior entomologist there, and we have agri-environment schemes in the UK, and I kind of oversee the pollinator based research. So do those agri-environment schemes, promote pollinator welfare, and the farmers kind of receive subsidies and things like that for it. But really, that's just my day job. So I've done that job for about 2 years. Prior to that, I worked for the National Bee Unit for 10 years as a seasonal bee inspector, and then as a regional bee inspector. So that's kind of like your apiary inspectors that you have
Right. So
in the
in the
US.
UK, National Bee Unit is basically there's one unified body that manages all the various apiary inspector activities.
Yeah. So we have two statutory notifiable diseases in the UK: European foul brood and American foul brood. Two statutory notifiable pests: small hive beetle and Tropilaelaps. And my job as a bee inspector was to go out and work with beekeepers and inspect for these diseases. And if they had diseases, then to sort of manage them and treat them. So we don't have small hive beetle in the UK, and we don't have Tropilaelaps in the UK. And this sort of work and this research started back in sort of mid 2022. As a bee inspector, we do what's called exotic pest surveillance, and we have central apiaries around the UK where we monitor for small hive beetle and we monitor for Tropilaelaps. And myself and Dan Etheridge, who's worked on a lot of the research, we're friends anyway, but we chat and we sort of say, you know what, these techniques that we use to monitor for Tropilaelaps, like we don't really think that they're terribly good, and nobody had actually tried them on colonies infested with Tropilaelaps. And I spoke to the head of the bee unit and I said, look, if I can get some funding, because it's the government and they never have money to do anything. If I can get some funding, can we go and actually trial these detection techniques on colonies infested with Tropilaelaps? And I think he just sort of said, yeah, you'll never get funding, but yeah, like just to kind of get me off the call and make me go away. But yeah, we did manage to get the funding, and four of us went off to Thailand. And we trialed the UK standard operating procedure for Tropilaelaps inspections. Found that like none of the techniques really worked, which was quite shocking.
You've just been wasting your time.
Yeah, so literally all of the samples we gathered, all of those inspections that we'd done had kind of been a bit of a waste of time. But we then radically changed and developed those detection techniques. We published a paper on that. But it was going and seeing Tropilaelaps in Asia and seeing how, so we went to Thailand, seeing how those Thai beekeepers dealt with it and the issues that they had with it. I mean, our sort of most enduring memory is that very first day we went to an apiary. We knew that there was Tropilaelaps there. The colonies were very small. You could see that they were being really impacted by this mite, but we couldn't find the mite. And we're very experienced. We do bee inspections for health and disease every single day, and we couldn't find these mites. So we thought, well, the reality of UK beekeepers detecting this when it turns up is quite alarming and probably pretty slim at this point. And then once we started working with the mite, there were just so many questions about transmission and survival and reproduction, and it was a lot of research has been done in the last couple of years by myself and many others, but back then there was very little known about it, and there were these big sort of gaps in the knowledge which are really important to deal with
questions
it.
that need to be answered for you to do your job, but that simply didn't have answers at the time.
yeah, and I think a lot of misconceptions, and I think a lot of particularly like the life cycle and the reproduction have been based on Varroa, and it's a very different mite to Varroa. It reproduces very differently, and a lot of the assumptions that we had. For our contingency response, so if Tropilaelaps turns up in the UK, what will we do? We would, for example, use treatments, amitraz-based treatments to treat it. Well, since and on paper that seems highly practical.
Right.
It's a miticide treatment, but what we've realized now is Tropilaelaps have this very short phoretic phase. They don't
Hmm.
actually if you use amitraz, they don't actually come into contact with that
Hmm.
treatment in the hives, so it's very ineffective. And actually
Right.
formic acid is much more effective,
Right.
but
If I rub an amitraz product onto a Tropilaelaps mite, I can kill it, but that's not happening
yeah.
inside of the confines of a beehive.
Yeah. So just we very quickly realised that the knowledge gaps were pretty significant,
Mm.
and then unfortunately, so that there wasn't really the funding within the National Bee Unit to continue that research. But myself and Dan Etheridge and Paul Davies and George Tong, so we were kind of the original group that
Mhm.
went out there, all bee inspectors. We wanted to continue that research. So I set up PHIRA-Science. It's a non-profit organisation, and just really to do this research. So it's primarily funded by beekeepers, beekeeping organisations, and we are all beekeepers. We've all sort of
Mhm.
kept bees for 20, 30 years ish, and first and foremost we're beekeepers. And then that sort of led us into the research side of it, and
Mhm.
finding answers that will be helpful for what was then beekeepers in Thailand and Asia. It's
Mhm.
since been detected in the country of Georgia and Western Russia, and they're having big issues with it. So solutions to help those beekeepers. And I think inevitably it will spread across Europe. Potentially it might make it to North America at some point. And
Sure.
and, and yeah, it's just all of those kind of important questions that need to be answered. Trying to do it before it's a bigger problem.
And how frustrating, I mean, how amazing that you've been able to create your organization, which can then take beekeeper donations so that the research can be done and we can actually have answers. And it helps a beekeeper in Thailand or Georgia or the United States, wherever the mites might show up, that basic science needs to have been done. So thank goodness it's happening. But how frustrating that you needed to create this whole separate, branch and funding structure just to be able to do it. That there hasn't been that gobs of money coming from the US or Canada saying, they're not
Yeah.
here, but if they did get here, we'd really like to know something about them. So let's actually fund some of this research. But thank goodness
Yeah.
you're doing it.
Yeah, and I think unfortunately, researchers the world over are in a sort of similar situation to me.
Right.
Governments the world over, the funding just isn't there. I just took a very pragmatic view of it, and it's like, right, well, let's find a solution. You can sit there and sort of go, okay, this is terrible, and it is terrible that lots of things aren't being properly researched. Or you can sort of PHIRA-Science was a bit of a long shot. It is just a group of four of us, and but we're very passionate about what we do, and everybody sort of works really hard at it, and I think that does sort of count for a lot. And I think the beekeepers appreciate that we're trying to get these solutions and answers, and be quite sort of practical and pragmatic about things, and I think I've kept bees for 20 years, and I very much consider myself a beekeeper before a researcher, if that makes sense.
Sure, sure,
So
absolutely.
yeah, it's that what's going to work in the apiary,
Right,
what's practical in terms of
right.
treatments, what's practical in terms of everything.
As a scientist or a researcher, you can get a notion into your head and say, well, that's sort of an interesting, alley that I could walk down with my research and maybe try to learn more about this. But as a beekeeper, you see yourself pulling a certain thread in the research and saying, you know what, this isn't going to lead anywhere useful. So I'm going to stop. Someone else can do this later. Some grad student in 20 years can do this. I'm going to
Yeah.
focus on, how can I keep these mites from killing my bees and somebody else's bees?
Yeah,
So,
and we
yeah.
spend a lot of time with beekeepers in Thailand and in
Mhm.
Georgia, and it's what do you need, and a lot of it is that seeing the firsthand experience of what they're experiencing, and
Yeah.
yeah, that's invaluable, I think.
Yeah, excellent. So we should mention that one of the reasons that we're here together is that a new book has just been published called Tropilaelaps at the Gate of Your Apiary. And this is a book with four authors, of which you are one of them. I will mention just sort of as a brief shameless plug that this book is published by the licensed beekeeping company, and that because we distribute their products in the US, this is coming to the US in winter of 2025 from Betterbee. So if anyone would like this book that we're about to chat about a bit, contact me, and we should quickly have a link underneath this video for folks to either pre-order it or to have it shipped to them. So can you talk a little bit about, how it is that this book all about Tropilaelaps, and I've had the privilege of being able to read through it. It is an excellent book. It is really what was it? I think Jeff Wilson from Canada said that it's sort of the gold standard as a guide, everything you need to know about a pest. So if you can just talk a little bit about your other authors on this project, and sort of how it is that the four of you came to be the authors of this book.
Yeah, so the book was originally started by Alex Uzunov, Marin Kobayashi, and Irakli Janeshai. So Alex is based in Macedonia. He works at Skopje University. He has been involved with honeybee research for 30 plus years, but again is also a beekeeper first and foremost. Marin, he's a third generation beekeeper, but again, so he's based in Croatia, and is similarly sort of involved with honeybee research. And then Irakli Janesia, so he is a veterinarian by trade, but he's kept bees since I think he was 12 or 13.
Oh, wow.
So he was the first person to kind of raise the alarm that Tropilaelaps was present in the country of Georgia. And they have worked on things like so the rapid brood decapping technique, which uses wax hair depilation strips to monitor for Tropilaelaps. So they were kind of separately working on various bits of Tropilaelaps research, and obviously myself and Dan
Mhm.
and my little gang were working on Tropilaelaps research. And Alex reached out to me. I obviously have sort of lots of photos and videos, and I share that on social media quite a lot. So originally it was, can we use some of your photos in the book? And I have, graphics and things like that. I was like, yeah, sure. Let's get the information
Right.
out there, and then yeah, it just sort of naturally evolved into, do you want to help? Just sort of you can write the kind of breeding section
Mhm.
and reproduction and things like that. So yeah, it just sort of naturally it evolved over the
Excellent.
summer, and and now we've all started working on sort of research together as well. So it's not just
Marvelous.
the book we've
Marvelous.
got.
Right, right.
Yeah,
The
yeah,
book brought you together, but you're still a bunch of beekeepers and Tropilaelaps researchers, and that's
yeah.
a dream team to get questions answered. And it really is remarkable that you're from the UK. We've got scientists from Macedonia, from Croatia, from Georgia, all sort of brought together. And I know in the US there are beekeepers in Alabama and Thailand who might never have met one another, who suddenly are now together because of this work. So there really is a tremendous opportunity for scientists to collaborate across these different borders and boundaries, which I think, if we have to find a silver lining, at least it's that we're forging these professional relationships as we fight this devastating parasite.
Yeah,
Um,
definitely.
so maybe we should just to make sure that everybody's up to speed. Can you just quickly tell us a little bit about sort of the origins of Tropilaelaps, where they come from originally, and then sort of where in the world the interactions between them and our honeybees have taken place that have led to these new developments.
Yeah, sure. So Tropilaelaps, they naturally come from Southeast Asia. So they infest the giant honeybee species that are native to Southeast Asia. So that's Apis dorsata, the giant Asian honeybee, and Apis laboriosa. So that's the giant Himalayan honeybee.
Mhm.
And
Which are sort of cousin
Tropilaelaps,
species, right?
yeah. So Tropilaelaps and these giant honeybee species have kind of co-evolved over millions of years, and the giant honeybees have developed this natural host parasite relationship, and typically a parasite doesn't want to kill its host. All it wants to do is reproduce and be spread around and kind of reach new areas. The issues began, so our western honeybees, Apis mellifera, are only native to Europe, the Middle East, and Africa. They were introduced into Europe by us at the end of the 19th, sort of beginning of the 20th century. Because they're very good at producing honey, and you
Mhm.
have these sort of bigger colonies, and you don't really have a honeybee species in Asia that is can be managed so easily, and it is so productive.
Mhm.
So Western honeybees were introduced, but unfortunately Varroa destructor jumped species from the eastern honeybee, Apis cerana, and that was the sort of first parasite to jump species. And
Right.
then Tropilaelaps also jump species from these giant honeybee species to Western
So
honeybees.
this is a recurring story, is that we've watched Varroa jump from from Apis cerana onto our bees, and then we've watched Tropilaelaps that has jumped from Apis dorsata onto our bees. So
Yeah, yeah,
it's clearly, it clearly suggests that there may be some problem in the original plan of let's bring Western honeybees to Southeast Asia and use them to make a little bit more honey per colony. Surely nothing bad could happen.
yeah,
Right.
exactly. I mean, I think probably lack of foresight back, back when people were moving these bees around, and then
Right.
unfortunately, so we then ended up with this pretty much unbroken chain of Western honeybees from Southeast Asia all the way over to Europe, to the
Mhm.
UK, and Tropilaelaps slowly started spreading through these colonies. And I believe that they first started being detected in sort of Pakistan, Afghanistan, which is outside of the range of these giant honeybee species.
Uh-huh.
So this is when they started sort of becoming out of their native range.
Which is
So
also
they
not a
probably
terribly easy spot to go do honeybee research.
no, no,
This is not a
exactly.
region of
Yeah,
the
no,
world that it's easy to just breeze in and say, I'd like to check all of your bees. There's
yeah.
mountains and wars and various issues that might make that a little bit challenging.
Yeah,
Mhm.
certainly. So that I think reports from around the sort of mid 1990s, something like that, and they've continued to spread westward. So there's unconfirmed reports in Iran. I think that they probably are in Iran, but the political situation there is somewhat
Complicated.
difficult,
Right.
complicated. Yeah, yeah.
Right.
Um, and then so it wasn't until they kind of reached Western Russia,
Mhm.
and so there's the Krasnodar region and the Rostov regions where it was confirmed, so early-ish 2024, and then late 2024 in Georgia, that really the sort of European beekeepers and North American beekeepers began to sit up, because that was considerably closer than the
Right.
previous known report of where Tropilaelaps had reached. And then this year, so the European Union reference laboratory put out a statement saying that it's unconfirmed, but there's potentially Tropilaelaps in Belarus and Crimea.
Mhm.
And I think part of my concern about these unconfirmed reports are the researchers did the work in 2023, and it's taken 2 years for that to be kind of
Yeah.
become more widely known. And because it's unconfirmed, it's really hard to get any information. So,
Mhm.
was it Tropilaelaps, or was it some other species of mite that was misidentified? If it
Mhm.
was Tropilaelaps, are they established, or did they die
Right.
out
Was
for
it
some
ephemeral
reason?
and then they vanished? Yeah.
Yeah. And and if it was Tropilaelaps and it's established, are they spreading? Because Poland, for example, is right next to Belarus, and
Right.
very very big beekeeping culture there, and bees are sort of exported from Poland and other countries
Right.
in that area. So quite concerning that there's just this black hole of information, and it's
Yeah.
very difficult to. I mean, they're a very difficult mite to find, so it's very difficult to get that information and find out exactly what's happening.
So, in the book, we on page 16, you have a map of all of the places where these, I guess it would probably be fair to say that that page is probably the most likely to already be inaccurate, and certainly the most
Yeah,
likely to become inaccurate over the next few years as they spread, as we learn more about where they are or aren't, and things like that. But it's so important for us to at least uh, you know, put the stake in the ground and say, here's where we think we know that they are and aren't right now. And it
yeah,
helps to identify that pattern of spread. But there is definitively a pattern of spread. It's not that these things have always been in Georgia and then they've just been noticed. It's that they genuinely were not there and then were there. And there have been real
yeah.
consequences to that, right?
Yeah, and so I've been working in Georgia this year a lot with Irakli, and The sort of anecdotal evidence is that they probably came from that Krasnodar and Rostov regions of Russia. So if you don't know Georgia,
Mhm.
there's the Northern Caucasus at the north, and then the Lesser Caucasus at the south. So it's actually quite an isolated country in terms of like biodiversity, and that it's quite isolated. But there is this, where the Black Sea meets the Caucasus Mountains. There is this land bridge, and that's what sort of people use to travel between the countries. And it's believed that Tropilaelaps came down from that northwest corner and probably arrived in 2022. So that's when beekeepers in Abkhazia started reporting these significant colony losses. And by certainly 2024, well, certainly 2025, it's right the way across Georgia, from over to as far as Tbilisi, which is on the eastern side of Georgia. But yes, it definitely wasn't in mid Georgia in 2024,
Right.
but it's certainly there now. So it's been frightening how quickly it has spread across Georgia. It's not a big country, but
Mhm.
the rate of spread has surprised me.
And that is very, it's very similar. Again, not to overly compare them to Varroa, but that's true of Varroa as well. Is that once they started spreading, they were spreading incredibly quickly, both country to country and over landscapes. Do you have a good sense, or do we have a good sense of how these mites actually spread from one side of Georgia to the other?
So there is quite a bit of migratory beekeeping in Georgia. So like I said, you've got the Greater and the Lesser Caucasus Mountains on the northern and the southern borders, and the center of Georgia is relatively flat, and that's where the majority of people live. In terms of beekeeping, so the beekeeping season, the colonies tend to overwinter down in the lowlands, and then the bees are sort of gradually moved up the mountain slopes, because things like we've got sort of linden and chestnut and various
Mhm.
other flowers sort of successionally bloom as they get higher up the mountain. So the beekeepers will move their bees up the mountains to utilize these honey crops. They probably get up to the high alpine meadows in sort of mid August. And that's when it's thought that they came into contact with other infested colonies from Russia,
Mhm.
and then the colonies are moved back down to the lowlands to overwinter in the lowlands. And
Got it.
it's that sort of act of moving your bees apart and then moving them all back together again, and that's what sort of
Right.
spread Tropilaelaps quite rapidly.
Which, as a beekeeper in the US, is obviously alarming, because that's very typical of US beekeeping, is that so many bees move so many miles in the United States to get from various pollination contracts or to chase honey flows, just as they're doing in Georgia. So there's a lot of similarity in that style.
Yeah.
In the book, though, you do also have that case study of the convent, where the nuns are keeping their bees. They're not migratory; they just have their bees there at the convent, and the Tropilaelaps showed up. So it's clear that, in addition to, I put my bees next to your bees, and your bees have mites and mine don't, and I pick them up, and then we both go back down to the lowlands. That's not the only way that they're spreading. It's also that they're able to spread sort of across the landscape.
Yeah. So the nuns are a very interesting kind of case study, and that's partly why we put it in the book. So so there are convent of nuns. They don't do migratory beekeeping. They've got, I think, about 86 colonies there. Very skilled beekeepers. And one of the nuns, Daniela, was actually a biologist before she became a nun. So
That's
I was just
handy.
there recently. Yeah, I was just there recently, and we took a microscope up to do brood biopsies, and she was thrilled. She was like right on the microscope and doing all the work for us. But yeah, so they didn't have any Tropilaelaps mites in 2024, so they were kind of aware of the
Mhm.
situation, and they went into the winter, and they did a lot of brood monitoring and checked their brood for Tropilaelaps mites. I think it was March the 3rd. I think they reported that they had one Tropilaelaps mite in one brood cell in one colony, and then another Tropilaelaps mite in another brood cell in another colony. So two infested colonies, each infested with one mite
Right.
in early March. I think they treated with amitraz at that point, because that was sort of assumed as a miticide treatment, it would
Right.
be effective. Um, And then so I was there in June and worked with them. And at that point, we checked pretty much all of the colonies, and all 86 colonies were infested. Some were very heavily infested. So through this season, so they've used a combination of queen caging. So they'll cage all of the queens in the apiary for typically about 30 days to make them all broodless. And so we initially thought, well, that would be very effective. If there's no brood, then the Tropilaelaps mites won't be able to do anything. But Irakli has worked with them, and they found probably within 3 weeks of releasing the queens from their cages, so that first brood cycle, that they were
Right.
starting to detect Tropilaelaps mites again. So they're probably, the assumption is that they're coming in. So foragers are either meeting in the wider environment, or you've got robber bees coming into the apiary. I mean, the apiary isn't sort of completely isolated, but it is up a mountain
Mhm.
in Georgia, and there's not that many beekeepers in the locality. So yeah, so then again by kind of mid-September, they were having serious mite problems. So we did some very strong formic acid treatments. And again, we got the colonies to a point where Tropilaelaps were undetectable in the colonies. But by sort of 1st of October, again, that reinfestation had happened, and the rate of reproduction with Tropilaelaps is so fast that you're again seeing between sort of 5 and 10% levels of infestation in these colonies. So it's this really difficult sort of seesaw balance between,
Mhm.
we seem to be able to quite effectively manage Tropilaelaps, whether that's with brood breaks or whether that's with formic acid,
Right. You can sort of
and get
knock
the
them
mite.
down initially,
Yeah,
but
yeah,
then
and get
they're
it
back.
to a point where they're not detectable in the apiary. And I think that
Mhm.
the first time, because we've done this as well with Irakli's apiary, and we were quite smug. We're like, great, we've killed all the Tropilaelaps mites.
Problem solved.
Yeah,
Let's get
problem
home.
solved. Yeah,
Yeah.
I'll go back to the UK, and
Yeah.
Irakli's messaging me sort of 3 4 weeks later, 6 weeks later, going, yeah, we've got a problem again. Like,
Huh.
what, and we obviously have research plans, so it's like, well, what do we do to kind of keep the colonies going until the next round of research starts? And it's that constantly sort of fluid problem solving that you have to do with it. So my experience this year has been, it's pretty easy to kill Tropilaelaps and control them, but the issue is that reinfestation is really high. So
Right.
whether they are
And
somehow
even
surviving, and
even one brood cycle after you've successfully controlled them, they're back. So it's
yeah,
not like you can
yeah,
buy yourself some respite. It's that it's sort of a continuous project.
yeah. So you're having to do something, whether that's biotechnical or
Mhm.
I'd say use formic acid, because that's the only thing that's actually penetrating into the brood cells. But you're having to do something every 6 weeks, 8 weeks, just to kind
Mhm.
of keep on top of it. And it's a lot of
Wow.
work for beekeepers. Yeah, once you kind of factor in the cost of that labor, let alone the cost of chemical treatments. Irakli has spent a lot of time in his apiary just finding
Right.
queens, caging queens, putting treatments on, manipulating colonies,
Mhm.
destroying ones that are really heavily infested, and so it's really changed the kind of landscape of beekeeping, and what you have to do to be able to keep your colonies healthy.
Mhm. And that's true really everywhere, whether it's Southeast Asian sort of style beekeeping or Georgian style beekeeping. It's that when they arrive, they radically alter everything, even in different sort of patterns of migratory, non-migratory, large hives, small hives, big honey crops, small honey crops. It's that they are such an insidious pest, and right in the brood nest, reproducing constantly as long as there's brood, means that there's just no opportunity to get away from them unless you've got a long brood break given to you by something like winter, and even that isn't enough to completely eradicate the problem, right?
Yeah, so again, with if we take the example of the nuns in the convent, so they caged their queens on I think it was the 15th of October, and didn't release them until the 15th of February. So you've got that long winter brood break, and like I said, they were picking up Tropilaelaps mites in March. So you can, whether it's a natural brood break caused by winter, or whether it's an imposed brood break
Mhm.
by the beekeepers. so yes, it kind of solves the problem for that period when the colonies are broodless, but
Mhm.
it's that reinfestation is very
Right.
quick, and then the rate of reproduction
Right.
is really high. So yeah, it's
And a lot of my own research on Varroa transmission, emphasized that as well, is that drift of bees from one colony to another is a great way to spread parasites. Robbing of resources from one colony by another is a great way to spread parasites. And you can even, I have video of a Varroa mite on a flower jumping onto a bee. And so, as you say, if two foragers bump into one another, it's a perfectly plausible chance for a parasite to say, you know what, I'd like a change of pace. I'm going to jump on this bee that smells a little different and ride her
yeah.
home.
Yeah, and Tropilaelaps are so much more mobile than Varroa mites. I mean,
Right.
when we work with them in the lab, we'll have brood frames, we'll be biopsying brood, and they will jump from the brood frame onto your hands and sort of, run up your arms and things like that. So we're always quite surprised, and I think that's probably, the heat coming from us. If you've been working on a brood frame for a while, it starts
Mhm.
to cool down a bit, and yeah, when Varroa
Suddenly you're a
just
little more appealing.
yeah, yeah,
Yeah.
and Varroa just sort of amble about quite casually, whereas
Mhm.
Tropilaelaps are on steroids almost. They're
Right.
just constantly running and very active, and yeah,
Well, even
very
the name, right?
agile.
Laelaps was the dog in Greek mythology that could catch any quarry that it hunted. So, the
Yeah,
speed has been recognized right from the get-go
yeah,
that these things are quick, in addition to
yeah.
being devastating. So you've mentioned a little bit about control of Tropilaelaps. We've talked about chemicals, the use of amitraz strips. You said are not terribly effective because they're not exposed. Formic you've mentioned formic acid as being effective against them. And I guess an advantage of formic is that it also kills some brood, which means that might also help to create that brood break. But otherwise, a beekeeper might have to rely on caging their queen in order to engineer a brood break in the hive. So are you finding that there are beekeepers that are able to use that strategy effectively, that brood breaks can be a good way to carry on making a crop of honey, even if it requires more labor, or is it at this point not viable? I guess are there Georgian beekeepers who are making do, or is everyone in the midst of crisis right now looking for more strategies or more tools to get themselves back to productivity?
Yeah, I think we certainly don't have a solution to control Tropilaelaps. So predominantly it has been those brood breaks, so queen caging, and like I said, the formic acid
Mhm.
treatments to control the Tropilaelaps. I think so one of the issues with certainly like beekeeping in the UK is that we're predominantly hobbyist beekeepers, and the thing that always sort of concerns me is that the even with the finding and the marking of the queens, it can be problematic. So
Sure.
if and with Tropilaelaps, you have to do these biotechnical control methods on every colony in the apiary, because if you miss one and there's any brood there, then you've got that drifting,
Right.
and the mites
The
are
mites
that
are right
mobile.
back in.
So
Yeah.
yeah. So a lot of Georgian beekeepers have been queen caging, but when you go to them and you talk to them, and it's like, well, I caged the queens in 80 colonies, but there were five that, well, where I couldn't find the queen, or they sort of made emergency queen cells, or
Mhm.
something happened and something went
Right.
wrong in the process, and brood was produced, and then they're almost sort of right back to where they started. But yeah, formic acid has worked well. I think we've trialed Formic Pro in Georgia, and we've also trialed some
I
Noravinca.
don't tell anybody involved in the inspection of international packages, but I may have been slightly involved in getting that Formic Pro over to Georgia.
Oh, there was no dodgy addresses used at all or anything
Right,
like that.
right.
No, no.
But again, that's the regulation has a hard time keeping up, right? When there's a product that
Yeah,
that very qualified, maybe the only qualified research team trying to actually tackle this problem, needs to get this product into a hive just to see how it works. And
yeah,
the paperwork is such a mountain that it would take, 2 years to actually get it there. So, sometimes
yeah.
sometimes there are reasons to expedite when there's a real dire need.
Yeah, well,
I think. I don't know if
I
that'll
went
hold
out
up
to
in court, so nobody tell anybody.
yeah, I went out to Georgia, and my suitcase was so there was a layer of Formic Pro, and
Mhm.
then several layers of wax hair depilation strips, and then various like timers and
Right.
but yeah,
If
microscope
a
parts and
customs
things.
official had opened that suitcase, they wouldn't have known what to think.
No, not suspicious at all. So yeah,
Right.
fortunately, I made it through without any kind of alarms going off. But yeah, yeah. But yeah, so we tried the Formic Pro, very effective at Tropilaelaps control. So we've trialed it in August, and we've also trialed it in October.
Hmm.
In August, the ambient temperature and the ambient humidity were very high. So ambient humidity was like over 80%, and the temperature was 25 degrees Celsius up to about 35 degrees Celsius. So
Got it.
really kind of at the limit of what you're meant to use formic treatments with.
Hmm.
So very good for Tropilaelaps control. I think we biopsied the brood after the treatments had been on the colonies for 60 hours. 99% of the Tropilaelaps mites were dead, and by like day
Excellent.
5, we couldn't. I think we found one Tropilaelaps mite in all of the colonies. And then there's Muravinka, which is this Russian treatment. So a much smaller treatment with a stronger concentration of formic acid. So
Also
that worked.
formic
I think that
acid.
was
Okay.
yeah. So that was like 85% effective. But
Mhm.
I think because it is a very small treatment, probably like 2 inch square little treatment. So I think
Okay.
just the and you sit it on top of the. I can share pictures with you, and it's
Yeah,
probably
sure. Yeah,
easier.
we
But
can put it into the video.
yeah.
Sure.
Yeah. So this Muravinca, it sits on top of the brood frames, and I think just the it just doesn't sort of dissipate throughout the hive as effectively as the formic pro is quite a big treatment, and it spreads across the brood,
Right.
and the Muravincas, so it killed some mites, but it wasn't as effective.
Right.
But
Less
the main
effective, but
issue,
maybe I could do it more often, or
yeah,
something like that. So
yeah,
there are these tools that have some effect. It's not like we're totally hopeless. If the mites showed up, we just throw our bees into the ocean and give up and all start raising chickens instead. It's that
yeah, yeah.
there's hope to be found here, but we just don't have that perfect recipe of how do I do the brood break? When do I treat with which
Yeah.
product to get to a stable position?
Yeah, and certainly when I went to Georgia in June and I met with beekeepers, and there's this was the middle of their first season where most of these beekeepers were dealing with Tropilaelaps, or perhaps they'd had it in 2024, and they kind of hadn't really realised what was going on, and this was the first season that they were kind of properly trying to deal with it. And certainly in June when I came away from Georgia, I was like, right, that's it. I'm just going to give up beekeeping because this is
It's
terrible.
hopeless, right?
Yeah, it's hopeless. But now at least I feel a lot more optimistic, and
Mhm.
there are these treatments, and yeah, we can kill them. It's not a
Mhm.
perfect solution, whether it's a combination of these biotechnical methods or formic acid. There will be a solution, and obviously, like the more we do, the more that will get refined, and
Right.
the better the situation will get. So
Right.
yeah, I think between June and now, I'm certainly a lot more optimistic, and hopefully,
Good.
if you talk to me in 12 months' time, then we'll be X amount further along with the research, and I'll be going, yes, use this, and
Yeah.
this is what you use.
And that's really the story that the book emphasizes is that it is a terrible but tractable problem, and that progress is being made incrementally. That was the
Yeah,
point that the Australians made for years about Varroa, was that they said, we know that they're going to get here. We know they're in
yeah,
basically every other country on earth. We know that they're going to show up, but every year that we can keep them out is a year where all of you have to continue your research to fight them. You all are going to start perfecting tools and methodologies. And so when they get here, we are going to have all of that advantage available to us. And I feel like the same is true here, is that the Georgian beekeepers suffering through this because their hives are so similar to the hives, that beekeepers in the UK or the US might be keeping, that style of beekeeping, those beekeepers facing this, but also the Thai beekeepers who have been fighting them even
mm,
longer, you know, every new product, every new test, every new technique gives us, another weapon to use when or if they do show up in your country
yeah.
or mine. So I guess maybe we can jump there, which is, what does a country do to try to keep Tropilaelaps out? Like, what is the UK doing to keep them out, and do you think it will work?
So I mean, I don't want to be the harbinger of doom, but I do feel a bit like that with proper layouts. But I think if you look at mankind's track record, we're very good at spreading all
Everything,
sorts of stuff, or
right?
everything all over the world. So and part of the work that myself and PHIRA-Science have been doing has been to try and raise awareness. So for example, in the UK, we've just probably about a month ago, we've just banned the importation of queens from Ukraine. So we were directly importing queens from Ukraine,
Mhm.
but yeah, and currently Ukraine can't establish whether their Tropilaelaps free or not. I think
Right.
the UK and North America, well, North America isn't an island, but obviously you have those oceans
Right.
between yourself and
Right.
Tropilaelaps. The UK is an island, so I think we have some things in our arsenal to help protect us from Tropilaelaps.
Mhm.
Mainland Europe, I think, unfortunately, it will just, if it's not just that natural spread across the continent, potentially there will be migratory beekeepers accidentally moving it to new places. It can also be spread on things like swarms and on shipping containers and things like
Mhm.
that. But for the UK, I think the UK does a pretty good job of trying to keep it out. So honeybee health in the UK is very well funded. Like I said, we have
Mhm.
the National Bee Unit, and beekeepers in the UK are predominantly hobbyists, and they're very vocal. They're very apt to, if they think that there's an issue, then they will contact their local members of parliament and write emails to the civil service and voice their opinion. So I think that's good. So in the UK, we have a system of central apiaries. So some of them are managed by the National Bee Unit,
Mhm.
where bee inspectors will go to these central apiaries 3 times a year, and that they'll do a full inspection for small hive beetle and Tropilaelaps, and they'll send samples back to the laboratory as well. So you've almost got that. If the bee inspector misses it, then they're checking brood, they're checking hive debris samples, things like that. For the presence of these.
And that apiary might be, next to a port or next to an airport or in a sort of a strategic spot to hopefully detect something early in its invasion should it show up.
Yeah. So there's a ranking of what are considered risk points. I mean, think things like docks and ports are top of the list. Queen
Mhm.
importers, that's very high up on the list. But in terms of small hive beetle, even things like plant importers
Sure.
are considered medium risk, because you can have the pupal form of small hive beetle in soils and things like that. And so we will have generally, it sort of depends on the ranking of how each place is risk, but we will have generally 2 enhanced central apiaries. So that's where the bee inspectors go 3 times a year. But there's also a number of voluntary central apiaries. So that is beekeepers that just live within, I think, 3 kilometres of these sites, and they are asked to monitor themselves. They receive training, and again, they send samples into the lab. So, and if you look at the map of the UK and the map of these central apiaries, it's pretty well covered, right the way around the coast, and then you've got
Mhm.
airports which are in the centre of the UK that are also monitored, queen importers, all sorts of different risks. So I think we do a good job, and the work that's put into it is very good and very important, because like Australia, keeping it out and delaying it for as long as possible is probably our best chance at
Right.
having some sort of manageable way to deal with Tropilaelaps
Right.
if it ever gets to the UK.
If they spread from Georgia across Europe, and they get to France, maybe the English Channel gives you an opportunity to delay further. Maybe all of these strategies help you detect and eradicate them. And even if they do eventually get established, hopefully that's in 20 years, and we've got so
Yeah,
many tools against them that there's just sort of a trivial problem by that point.
yeah.
Got it. Is there any, well, I guess let me ask you this because this is always a question of interest to me. Why is it that they are so harmful to Western honeybees, but they are not that harmful to Apis laboriosa or Apis dorsata, to the original host? Do we know what it is that those bees are doing that is keeping the mites from being so devastating?
So Apis dorsata and Apis laboriosa, like they're pretty under researched, and that's
Mhm.
understandable because they nest really high up in
The
trees. Like
laboriosa,
they're
you have to break your neck just to look at one, let alone get up onto the cliff to see one.
Yeah. So they're not easily accessible. They're big bees. Like
Mhm.
it's a lot to deal with. But the work that has been done, so just their natural biology, like I said, they've co-evolved with Tropilaelaps over millions of years. And so Apis dorsata, we tried one experiment to see how long Tropilaelaps could survive for on adult Apis dorsata, and the bees themselves actually killed the mites within sort of 72 hours. And as soon as we introduced the mites onto the bees,
Mhm.
they knew we'd put these mites on them. So there's lots
They
of
started
alarm
grooming.
pheromone. Yeah, lots
Got
of
it.
alarm pheromone release, lots of buzzing. They all started grooming each other. Some other researchers have done similar work, and they could see that the dorsata had like bitten the mites, they'd bitten their legs off, things like that. So they'd actually caused them damage. So one of their natural responses is this grooming mechanism. They also migrate each year, so they will typically spend the dry season. So I've worked a lot in Chiang Mai in Thailand. So for those colonies, they will spend the dry season in and around Chiang Mai, which is a bit sort of lower down. When the raining season comes and that forage dries up in the Chiang Mai area, they then migrate up into the mountains. They probably travel for about 130, 150 kilometers to sort of reach these new sites. And when they migrate, so it takes them several weeks to kind of migrate up into the mountains. They're broodless, so they don't
Right.
build any comb, they don't have any brood, so they themselves are broodless for
Mhm.
several weeks, months at a time. Nobody knows what Tropilaelaps do, but the natural host has this broodless period, and Tropilaelaps does something to be able to survive and with them. And then just things like, so Apis dorsata, even though they're a bigger bee, the sort of development time from egg to adult bee is shorter. So
Mhm.
that means that Tropilaelaps have less opportunity to reproduce. So if they get mellifera, they've got this long, it's a bit like Varroa preferentially infesting dronebreed, because that takes longer to emerge and hatch out. It allows more mites to
Right.
develop.
There's just more value in
So,
going into a drone cell because it's going to be capped for longer. And this is even what we see with Varroa resistance in African bee subspecies. We find that Apis mellifera in Africa tend to have shorter development periods,
yeah,
and that seems to be part of how they're able to manage having Varroa populations better than populations like the ones I've gotten in the US that take their sweet time of 3 weeks to
yeah.
produce a new worker.
Yeah. So the giant honeybees, they've got these natural defenses, but
Mhm.
when Western honeybees were introduced, they're not naturally infested by any parasites. So they're not naturally infested by Varroa. They're not naturally infested by Tropilaelaps. And say Western honeybees have been in Asia for say 100 years, so they've only had 100 years of evolution with these parasitic mites to try to develop some sort of coping mechanisms with them, and you've got dorsata that's had millions of years to kind of co-evolve and develop these things. So when we've done things like we've introduced Tropilaelaps onto Western honeybee adults, they just don't have that grooming behavior. They just can't clear the mites from their bodies. And when you sort of see them on the comb, and that the bees tend to sort of not overly react to them, and certainly not in the way that dorsata do when you're introducing the Tropilaelaps mites to them. So they're just this naive bee species that hasn't had the opportunity to develop any kind of mechanisms for dealing with them, and that's why the Tropilaelaps can feed on the honeybee brood. So whether it's Apis dorsata or whether it's Apis mellifera, they can feed and reproduce in the brood. It makes no difference to them. And that's kind of how we've ended up in that situation. It's just poor old Western honeybees just can't unfortunately cope with them, and they don't have those strategies to deal with them.
Right. But I guess obviously our bees are naive and therefore suffering greatly. But we've seen the same thing with Varroa, with tracheal mites, that something like grooming behavior is found in Western honeybees, just not with quite the same behaviors or the same intensity that might be needed. So there may be a possibility that there are some Western honeybees that can groom better, and if we breed from those, that there might be some opportunity to start to cultivate natural resistance in our bees. So that's unproven, but at least sort of a hopeful prospect that over the course of the coming, years or decades, we might be able to start producing a more Tropilaelaps resistant Western honeybee, should we all need to do that.
Yeah, and I think certainly when Varroa arrived in the UK, there were very big colony losses. It was that naive bee population
Right.
to Varroa, but now 30 years down the line, we are getting those resistant traits coming through, and
Mhm.
the honeybees themselves are dealing with Varroa much more robustly. So yeah, I think it will be a process, and certainly what we're seeing in Georgia is it's that initial naive population of bees that are really struggling to cope with these
Mhm.
mites, and hopefully over time they will start to adapt and develop
There'll be
mechanisms
some
to deal
bounce
with them.
back. Yeah.
Yeah.
Which again just emphasizes the importance of keeping them out of the UK as long as possible, keeping them out of the US as long as possible, even if it's inevitable that they should show up. We would want to make sure that we've learned more about what can be done. Maybe Georgian beekeepers will produce a Tropilaelaps proof bee because they'll
Yeah.
breed all of these great genetics into them, and then we can just spread those bees around the world before the mites show up. So there may be strategies to be found. I guess I should mention before we conclude here, just on this front of how or if we can keep them out of a region. So we're recording this on October 28th, 2025, and last Tuesday there was a live stream widely watched across the U.S. where some folks, some beekeepers, were discussing reports of a discovery that had been made and managed by the appropriate authorities of a ship. As the story went, a bunch of Apis cerana were found on a ship 36 miles off the coast of New Jersey. And, why aren't they telling us anything? And what's going on? And what are we going to do when these mites get here? Well, a few days later, New Jersey, finally released their official press release, which said that it was actually 3,600 miles off the coast, which is about as far off the coast of New Jersey as London, England is. So it's not quite as close as it had been led to be believed, but it was also Apis dorsata. It was the original host species that had some Tropilaelaps on them that had been found on this ship. The folks on the ship recognized that moving bees from country to country was not so good, so they contacted ahead of time. They said, "Hey, New Jersey, you're our next place that we're going. We're on our way to you. What do you want us to do?" They followed all the instructions. The bees were caught and killed, frozen. The mites were eventually identified, but to some extent, that shows that we have an immune system for biosecurity to protect our bees in the U.S., and the immune system worked, right? Like we didn't have the problem because we caught everything ahead of time and responded appropriately. So, I'm sure I don't know that the UK has had direct near misses like that, but I know that around the world, everyone trying to keep these mites out, there are systems in place to monitor for them. There are systems in place to exclude them. Do you think that there's more that needs to be done? From your perspective, from your position, do you think that countries need to be more proactive in some way about this, or do you think that using the systems that we've already got in place will perhaps be enough to slow that spread, to delay that spread of them, so that we can continue our research on how best to kill them when or if they show up in our apiaries?
Yeah, I mean, I think, first off, like hats off to everybody on that ship that kind of recognized,
Right.
what they'd seen, and they clearly followed a procedure that was set out. To everybody that worked for the US government that went out and dealt with it, and excellent work. It was handled in the best possible way, and that's a real credit to everybody that was involved in that. I think. I think my position is like we can always do more. Like
Mhm.
like if there's the resources to do more, then we should probably do more, because while that Apis dorsata colony was caught and reported, and the proper procedures were followed, we are only human. Like people do make mistakes, and these boats with shipping containers on, there's lots of crevices, there's lots of nooks and crannies. It's not just Apis dorsata and Apis cerana that can transport Tropilaelaps. Apis florea, so that's the red dwarf honeybee,
Mhm.
can also spread them. So we've had Apis florea arrive on a shipping container in the UK. I know that Malta currently has Apis florea. So these other honeybee species do get around the world. Like they are
Mhm.
transported on boats and planes,
Right.
and
Once
goodness
again, it's
knows what.
humans' faults for moving everything everywhere, right?
Yeah, and with the best will in the world, I genuinely don't think that we're going to find every honeybee colony that enters the UK, the US,
Mhm.
on a plane, on a shipping container, on whatever it is. So I think certainly the central apiary system that we have in the UK is very good,
It
and
seems like an ideal model to try to identify, especially in places where you have volunteer beekeepers who might be able to be drafted into the program, apiary inspectors, various programs that could fund, support, and continue that monitoring, so that at the very least we can identify things soon after they've happened, and not, 6 years later, everyone looks around and says, "Why are all of our bees dying? Oh, it must be these tiny little
yeah.
mites."
Yeah, and the thing is, we don't want to be so in the UK. Varroa was found in April 1992, and
Right.
by October 1992, it had then been reported, right the way
Right. They were
across the UK. So we
ubiquitous.
don't, and it's thought that it had been in the country for at least a year, probably longer, before beekeepers
Mhm.
kind of realised that. A big part of the work that we've been doing in the UK is raising that beekeepers' awareness, and we've made a series of videos on just simple ways that you can monitor for Tropilaelaps. And I'm constantly encouraging beekeepers here to make these techniques, so things like rapid brood decapping, make them just a part of your. Not You don't have to do it every bee
Mhm.
inspection that you do, and you don't have to do it on every colony. But if you can, once a month, once every 2 months, pick 5 colonies out of your 20 colonies, and just do a quick check, because I firmly believe that, when it gets here, it'll probably be a beekeeper that, spots it in the US, spots it in the UK, and the earlier we can find it, the better. The
Right.
best chance that we have at dealing with it.
Right.
And yeah, so I think whatever the US government can do to support beekeepers and to support this monitoring and enhance central apiary surveillance, I don't think any of that can be a bad thing. I mean, obviously, money is an issue, and it
Mhm.
always is, but you've got to look at the impact that Tropilaelaps will have to North American beekeeping if it gets there, and
Right.
wider agriculture and crop pollination.
Right. A small
It's going to be a
investment
big
per
deal.
year to keep them out is probably worth it versus the massive cost of them arriving.
Yeah. Somebody needs
Yeah.
to sit down and do the maths and go, okay, actually, like if it
Right.
gets here, it will spread very quickly. And certainly initially, while you've got a naive bee population, it will do a lot of damage. Yeah, American beekeeping will recover eventually, but it's that interim period where you're trying to sort of deal with things, and people's livelihoods depend on it.
Right.
So yeah, I think do as much as you can to keep them out, and put as much resource as you can towards that, and hopefully science and research will have evolved enough that when it does get there, we can go, well, you can just use this treatment, and it's quick
Right.
and simple, and it's not as big an issue as it currently is, and we're not in a Georgian beekeepers situation where
Right.
we're able to manage it.
It's an unfortunate problem, but it's not a catastrophe like it has been for some
Yeah.
of these places that were just completely naive. And I really think that just to plug your book one more time, I think it really is such a stunning example of how much researchers like you have been able to learn in such a small amount of time. That really that this is like most of these, just like Varroa. Varroa was sort of a throwaway report of some new mite species on Asian honeybees when it was first reported in what 1912. But suddenly it
Yeah.
turns out that when they get onto our bees and start causing devastation, there's a lot of things that we never bothered to learn about them. And
Yeah.
so, I think the book is an excellent highlight of everything that we know, a great deal that we do currently know, and also it's quite easy while you're reading it to see those gaps. And you guys don't hide them. You say this is a thing that we need to know. This is
Yeah.
a thing that needs to be learned. This is a thing we're currently studying, or
Yeah.
that we will have more data on next year. Um, so if someone wants to cultivate some expertise in these mites, to understand a little bit more about what is being done, what we know about them and what we don't know, I really think the book is it's very accessible. It's something that a scientist can read and get a lot out of, but I think it's something the average beekeeper can read and get a lot out of as well, because it's cutting edge information presented in a very clear and accessible way.
Oh, thank you. Yeah, I mean, we want beekeepers to read it. It's important that beekeepers are educated, and
Right.
having that knowledge, it can't be a bad thing. But thanks, David.
Yeah. Well, thank you so much for taking some time to talk to us. I don't want to distract you from ongoing and important Tropilaelaps research, so I'll let you go. I will point out that this coming January, you are one of the speakers at the North American Honeybee Expo. We're going to be there as well, and so we'll have a few copies of your book, and perhaps you'll swing by the booth and you can sign copies for people who want to pick one up, something like that.
Yeah, sounds
All
good.
right.
Yeah, sounds great.
Well, terrific. It has been a pleasure talking to you, and I hope people have really enjoyed learning from Maggie about all of these marvelous, research projects that have come together in this time of urgency and crisis to try to learn more about Tropilaelaps. Thank you very much, Maggie Gill, for joining us, and hopefully we will hear more from you. Hopefully it'll be that you've defeated Tropilaelaps and you're on to studying, something nice and easy like honeybee nutrition or pesticides or something like that. Thank you so much for joining us.
Brilliant. Thank you.